welcome to This Explainer. Today, we're diving straight into the complex, sometimes messy journey of genomic data. If you're a research scientist trying to wrap your head around a solid background framework for the next generation sequencing, or NGS, bioinformatics pipeline, well, you're in the exact right place. We're going to explore exactly how raw data transforms into targeted, real-world clinical action. So think about the sheer volume of data pumped out by modern sequencers. We're talking about a massive sea of billions of raw A's, C's, T's, and G's. Our big overarching goal here is transforming that intimidating chaos into a single, actionable clinical signal. How on earth do we find that one tiny needle in a billion-piece haystack that could actually save a patient's life? To answer that, here's our roadmap for today. First, we'll look at the precision medicine pipeline as a whole. Then, finding the signal through secondary analysis. Third, we'll dive into the genomic knowledge ecosystem. Fourth, targeting critical oncogenic drivers. And finally, making that ultimate clinical decision. Let's get into it. Section 1. The Precision Medicine Pipeline. Okay, taking a 10,000-foot view of the pipeline, just notice the vertical progression here. We start way up at the top with secondary analysis, moving from raw sequence reads down to variants. Then, that middle layer is annotation and database integration, which is really where we add the necessary context. Finally, the bottom layer is clinical interpretation and actionability. It's essentially a giant, continuous funnel distilling massive amounts of raw noise down to highly targeted clinical insights. Moving on to Section 2, Finding Signal Through Secondary Analysis. Let's actually break down this computational progression, step by step. In Step 1, those raw FASTQ sequence reads are aligned to the human reference genome using tools like BWA-MEM, which creates our analysis-ready BAM files. Then, in Step 2, our algorithmic spell checkers, specifically GATK4 and MUTEC2, step in to identify somatic symbol nucleotide variants and indels. They do this by comparing tumor samples directly against normal controls. Finally, in Step 3, these isolated variants are packaged up neatly into a VCF, or variant call format file. Just like that, we've successfully gone from raw strings of letters to a highly focused list of mutations. Now, as you probably know, processing these massive data sets takes immense computational time. I mean, it's a huge bottleneck. But consider the scale of optimization that's actually available to us now. Compared to the standard BWA-GATK workflow, high-performance alternatives, like Sention, for example, offer up to 10 times faster alignment and a completely staggering 20 to 50 times faster variant calling. When you're dealing with large research cohorts, this kind of speed optimization isn't just nice to have, it's absolutely game-changing. Which brings us to Section 3, the genomic knowledge ecosystem. So here's the thing. A mutation all by itself, it's really just a data point. We desperately need context. Enter the genomic knowledge ecosystem. Think of this as a massive, peer-reviewed brain that cross-references our tiny variant against literally decades of scientific literature. We rely on foundational resources like COSMIC and TCGA, alongside ClinVar for human variation. But the real clinical power, that lies in evidence aggregators like OncoKB and Civic. These platforms provide curated assertions based on actual drug approvals. This whole ecosystem basically surrounds our raw variant with the total sum of human genomic knowledge. Next up, Section 4, Targeting Critical Oncogenic Drivers. Over 165. That number represents the staggering scale of modern precision oncology right now. Knowledge bases like OncoKB currently track over 165 therapeutically actionable genes. Think about that. Out of thousands and thousands of potential mutations, these are the specific, proven targets where an alteration can actually direct a real medical intervention. And tied directly to those actionable genes are over 130 associated drugs. So our interpretation zeroes right in on these critical oncogenic drivers. We're looking at things like KRAS for therapy resistance, BRCA1 and 2 for DNA repair, and TP53 for tumor suppression. This step is absolutely crucial because it connects our cold computational data directly to living, treatable biological pathways. Finally, Section 5, the final clinical decision. Okay, to translate all this complex bioinformatics into clear, actionable clinical language, we categorize our findings into 4 distinct tiers. Tier 1 represents strong clinical significance. These are your FDA-approved therapies. Tier 2 indicates potential significance, backed by well-powered studies or clinical trials. Then there's Tier 3, the tricky one. This is unknown significance, or VUS, where the data might be conflicting or completely new. And finally, Tier 4 represents variants that are benign or likely benign. It's a really elegant framework that strictly separates your Tier 1 certainties from your Tier 4 benign noise. And here we are. This final clinical report is the culmination of the entire pipeline. It proves that the immense, almost overwhelming complexity of raw sequencing data, all those billions of base pairs, the intense computational alignment, the whole shebang, can actually result in life-saving, actionable precision oncology decisions. We've successfully mapped that incredible journey from a raw FASTQ file all the way to a targeted therapy recommendation. I want to leave you with this final thought. This pipeline is not static. Our genomic knowledge ecosystem is growing every single day. As those actionable gene databases continue to expand rapidly, how will the speed of our alignment algorithms, like those high-performance alternatives we discussed earlier, keep pace to actually deliver tomorrow's cures in a timely manner? I really hope this explainer has clarified the incredible journey of genomic data for your own research and maybe sparked a few new questions about the future computational limits of precision medicine. Keep exploring and keep questioning the data.